Hey, I'm Jake.
I'm Jillian.
I'm Bella.
And I'm Emma, and this is our floating disc lab.
The equation for photosynthesis is 6CO2 plus 6H2O yields C6H12O6 plus 6O2. Plants, algae, and photosynthetic bacteria take in light from the sun, molecules of carbon dioxide from the air, and water molecules from their environment and combine these reactants to produce food. A plant has different structures in order to perform different tasks. Leaves take in sunlight and CO2 from the air. Roots are planted in the soil to take in water and minerals. The stem contains microscopic tubes that transport water, minerals, and food from the roots and leaves.
The group concluded that if we altered our variable by tripling the carbon concentration of the solution within the experiment, then photosynthesis would occur at a higher rate than the initial baseline because there would be more carbon to be turned into glucose. Materials used within this experiment were 300 milliliters of 0.2% bicarbonate solution, a glass beaker, a petri dish, regular water, spinach, a hole puncher to acquire leaf discs, a syringe plunger, an incandescent light, and a glass beaker. As we contained all the materials necessary to have an accurate and successful experiment, we also had to calculate certain variables in order to visualize the effects of tripling the carbon concentration. The independent variable within this lab was the triple bicarbonate solution, as its variation did not depend on another variable throughout the lab, and it was the variable that changed in order to see the effects on the rate of photosynthesis. The dependent variable and the variable that values dependent on another was the rate of photosynthesis as it was tested and measured within this experiment. The constant in our lab was the temperature of the water and the temperature of the spinach, as it was kept the same when we tripled the carbon concentration.
The purpose of creating a vacuum with the syringe is to remove all air pockets from the leaf discs and replace them with water. While the discs were in the bicarbonate solution, they received carbon dioxide needed for photosynthesis from the bicarbonate water. The leaf discs were able to float due to their exposure to light, which split the water molecules present in the leaf, creating oxygen that made the leaves buoyant. For our experiment, we altered the photosynthesis equation by increasing the amount of carbon present in the reaction. One possible source of error is that our baseline data did not provide sufficient data for us to compare experimental data with. In this control data, the leaf discs would not float when submerged in the bicarbonate solution. Another source of error is that the water from our heat sink was dropped into the bicarbonate solution while collecting our experimental data. This could have slowed down the rate of photosynthesis.
Comparing our data, it was obvious that the rate of reaction increased when experimenting with our variable. Within 10 minutes, all 10 experimental leaf discs floated to the top of the solution, while for the control, after 10 minutes, there were no floating leaf discs. The data collected from the control and experimental supported our hypothesis that the triple concentrated bicarbonate solution would increase the rate of photosynthesis. It is understood that if you increase the concentration of reactants, then the rate of reaction will increase. Another group performed this experiment, comparing the rate of photosynthesis of refrigerated and room temperature spinach. Their data shows that the room temperature spinach had a quicker reaction rate of photosynthesis. This could be due to the delayed effect of the heat on the refrigerated spinach, not separating the water molecules as fast.
Thanks for watching.
Can we please have 100?